Hello, everybody. My name is Kevin Kocek. I'm the founder CEO of Chemical Q Device. This is event seminar 130 for drug discovery generative AI using Tensor Network GPT or BERT. So what's on this cover is basically all the different things of transformers that can be modified with tensor networks. So I'll get to that later in the discussion. But everything from the inputs, so the embeddings, multi-head attentions, fully connected layers, these things are addressed to make the model a lot smaller, so compress it, and then also to make it more explainable, along with other things as well. So you'll see those kind of highlighted in those black boxes. So if, depending on where you're coming from here, on the left is basically a rough overview of GPT, and basically you see that it's unidirectional, so these arrows don't come back down. And then on the right, and it stands for Generative Pre-training Transformer, it's used for generative artificial intelligence, meaning creating new things based on what it was previously trained on that's not labeled typically. It's in an unsupervised manner. Now with BERT, it's a little bit different. So BERT stands for Bidirectional Encoder Representations from Transformers. It's more versatile for tasks requiring this understanding of context and relationships within a given text, but it lacks the generative capabilities that the GPT does, i.e. that's what the G stands for in ChatGPT and other GPT models as well. So this is drug discovery. So drug discovery is a very broad field. I'm focusing on the amino acids, or like the protein generations right now. And if you ever took biochemistry, this is like the first week that you have with all the different amino acids. So they all, except for proline of the 20, they all have the bottom structures the same. So if this brings back, recollection of organic chemistry, this is good. But basically, these are individual amino acids, and they condense, meaning that it removes water when they come together into small peptides and then keep growing into these bigger polymers called proteins. Now, the top substituent is what is different in all of these. So if you look at the glycine is the smallest molecular weight, 75. Alanine is bigger, so a methyl group, so CH3. And then you start getting into protons or hydrogen atoms that are more acidic, such as serine. pKa is 16, that's still pretty high, but a thiol group, an SH here, is more acidic. So the lower pKa, the more acidic it is. So these are used all the time. So these are the common known amino acids that they're going to be used a lot in drug discovery. You'll see other ones mentioned as X. So X is some other amino acid. Sometimes it can actually mess up your code. So if it's trying to put X in, but the model that you have doesn't run it, I had to do that at least once in fixing that. Because X was not recognized. So these are 20 different letter codes. They're all single letter codes that are assigned. So glycine's G. Say, for instance, down here with the amines, asparagine is N, glutamine is Q, okay? So you'll see these one-letter codes all over the place for drug discovery. And Gen AI is a huge thing. You can see this everywhere from Stanford Medical and other sources too, that using this, super powerful GPUs, not all of the time do you need them, but, for the big applications. And obviously my in is with the tensor networks and, basically to make them more efficient. So there's a bunch of different resources out there. So this first one I actually put up on LinkedIn, and it's using, so the way I'm categorizing this folder in my, in the GitHub is by models. Okay, so a GPT-2 model is originally trained, that's the GPT-2, but a GPT-2 is called fine-tuning on some other data set. So in this case, it's the entirety of the proteins out there are pretty much close to that. And so basically what was done is that I put in a bigger sequence. So and I showed you the slide before. M, S, K, K is lysine, I believe. G is glycine. Say, for instance, these are all the different combinations of those 20, right? So in this spot, it's M, S, and then you look over here, it's P, N, and K. Okay, so for each spot, it's one of 20 letters. The letter X, I didn't include in this because it's messing things up. And then the other 5 are part of the alphabet that aren't assigned to one-letter codes for amino acids. So again, We're over here. So if you're coming from this biochemistry point of view, DNA to RNA to protein. So we're basically, over in the protein side, and then the proteins are made up of individual amino acids, as mentioned before. Now, the cool, fun, and exciting thing with all of this is that PROT-GPT-2 will likely move up to like a GPT-3 or 3.5 as time goes on with bigger models and more trained parameters. And then you can fine tune on additional protein data sets that are, say, more corrected or bigger or better in some type of functionality. And the thing is with these, and you could use these pipelines through Hugging Face. And the specific one is called text generation, is very, very popular. So this is the same thing as like if you were to type into Chat GPT 3.5 or 4, it's using text generation. And in that specific case, it's GPT 3.5 or 4. But in this specific case, because we're looking at proteins and we want the data that is most associated with being, amino acids and how they relate in protein chains, where the author's name is nFerris, and then it's GPT-2, okay? So it's one of OpenAI's open source model that it's based on, and then nFerris went in, and then, basically with the protein data set, and I'll get to what exactly those were too. Now, the generating the sequences part, and again, this doesn't have to just be amino acids and proteins. The max length in this specific case is not necessarily the number of amino acids. I think it has more to do with tokens. So there's other modifications that you could do. So the original model, and a good place to find starting code, by the way, with this is on the model cards on Hugging Face's sites. And you can just copy and paste that code, and you cite that you got it from this, model card. But it only tends to be just bare functionality. So I took it a little bit further. Of the 10 generated sequences, I took the specific one and compared it to an original reference. So actually this up here is the reference, and then this generated is here. So it says about 29-30% similarity, between the two proteins. And then the reference molecular weights are different, and then percent similarities in molecular weight 63. Now, the isoelectric point is basically that break-even point where it's neutral of the whole entire protein. So you're going to have amine groups, which are basic. You're going to have acid groups like carboxylic acids, which are acids. And so that's going to bring it down, and the amines are going to bring it up. So the two different isoelectric points for the reference for this reference sequence up here, which could be anything, it's just a reference, and was 9. And then this bottom one is 12, so more basic. And the percent similarity between those two is about 66%. So this was done for similarity, molecular weight, isoelectric point, to get this additional detail. And it's really important, even if it seems trivial, to work on these things like, oh, I know those things, I know what all these things mean, is to work on it with the computer programs to progress through that. So this next one is basically another PROT-GPT-2. So if you're new to Hugging Faces, you're going to go over, if you wind up coding for me, is that PROT-GPT-2, it has all of this. So it's text generation, which shows up, see how it has to be in the specific format, lowercase text generation. Now, when you copy and paste this, nFerris, it goes right into model, okay? And then you can't just use PROT-GPT-2. And if they're similar types, a lot of times, like I'll show you in an upcoming one of the research that I was doing, is that you could just copy and paste the cell, and the only thing you have to do is change the model, as long as it's the same type. Like if it's, fill mask versus text generation versus text to video, text to image, image to text, so on and so forth. So this specific one, I generated, in addition, these perplexities, and it's basically a different, so it's going to generate these 10 sequences here. So when it says number of return sequences, it generates 10. And then I chose 2 of them, sequence 1 and sequence 3, took the difference, and then get this number. And it's a rough thing. You could also see when you generate the actual numbers, whatever number is lower in perplexity in this case, you would tend to use that one. And then this is ProtBert. So this is different. So remember how I started off talking about GPT, generative, pre-trained transformers. Now in this specific case, it's BERT. It's this bidirectional. It's not generative. So, but it could do a lot more, the different distinguishable tasks. So you can make the argument either or, which does more, GPT or BERT. If you're just doing generative, it's obviously going to be GPT. But I think as far as the wide, assortment of different problems to be solved, it sounds like it's more with BERT, but it's not generative. Okay, so this top figure here is basically an example. It says the pink amino acid residues are all cancer-related single amino acid variations. So this makes it think like, okay, well, this is probably some mass fill, right? So a lot of times you don't, if you're working with databases, there'll be just a missing amino acid. So, or you want to do other research with that. So this is by Liu et al. In 2021 in Scientific Reports. It's a Nature paper. And then you'll find this. So both ProtBert and the GPT-based Prot are both, featured in papers. This one, like I said, it's Nature paper. I think the other one was more archive. But that doesn't mean it's a bad thing, because a lot of times you just want to get it out, get on with your life, into a repository. So what was done in this specific thing is I inserted 3 different masks in this specific one. So instead of putting this, instead of printing out this big long sequence, it just puts the gaps in between the masks as UNKs. So probably unknowns, right? As far as how it's printed out, but it is unknown, but it is known. Okay, so here's a mask, there's a mask. And then in this specific way, I told it to put X or some amino acid, and it said if putting that in, it's a close score to 1, as it should be, because if you're representing something that could be any amino acid in this specific place here, the score should be 1. Whereas the other ones were much lower, and I didn't think until after I did this as far as which to include. But if it's something like if you want to put a lysine in like a K or proline for P, those types of things, the value, the score is going to go way down. Okay, so I think it's a very important thing in any type of research. You try to work with one variable at a time. So if you have 3 masks, or like basically 3 amino acids that are not known in this specific protein sequence, I think there is a way to generate, based on, so all of the answers, this 99%, it's based on how well it was trained on the original, protein data set. And of course, these things are always being improved, and they actually recommend to fine tune on top of that with your own, protein data that's more similar to what you're working on, not like the broad collection. But nevertheless, in this specific one, it printed out 30 different combinations. So, if I'm printing out 10 each and there's 3 masks, then say for the first one, it's going to, take out the mask, this value here for all the 10, and then the next 10 that prints out is going to take out and put a value in for that mask, right? So unless it's some technical restriction as far as doing all 3 at the same time, which is what I was trying to do, that's probably an option too. So basically a lot of this uses PyTorch. Hugging Face is not on its own. You have to typically use PyTorch, TensorFlow for most of these, or TensorFlow for most of these tasks. Okay, so are there any questions at this point? So that was a lot of the kind of the biochemistry, the drug discovery. I touched on the generative aspect of GPT in running these models, and I touched on the fill mask, which is available to use with BERT. So again, if you go to do this, a lot of times you can't do everything with GPT, and I think there's stable diffusions in the mix here too, and you can't do everything with BERT. So it's a lot of times like, what kind of thing do you do? And I would say the industry is just moving very fast towards generative AI, but there still are going to be tasks. So actually in this next set here too, and feel free to put any questions into the chat as well, is going to focus on some of these other ones. So stable diffusion, and I'm going to go into explainability with tensor networks. And then this last part here, so distilled BERT is based on BERT. And then there's also BART, which is like BERT, but I think it's also autoregressive, like a variant of BERT, GPT, Starling, Hermes, Mistral. So these 3 here, I believe, are either stable diffusion or GPT based. Sometimes it's a little hard to tell. So what was done here is basically I gave it a fragment, and my fragment here is medical generative AI is very, and the top hit actually came from, this is a GPT-2 large, I believe. It's in the notebook. It's up on GitHub. And it says medical generative AI is very different from the traditional AI approach. It is a system that learns from experience and it adapts to the environment. It is not a system that is programmed to solve problems. It is a system that learns from its environment okay, so I'm not quite sure if it was a character limit at that point. But basically, I can put it head to head with GPT-2 small, GPT-2 medium, large, and XL. Like I said, I think in this case, I believe this answer came from large, right? So by and large, I would say the models that are up on Hugging Face that I've used so far would likely need some other type of fine-tuning with your own data compared to a more streamless experience with ChatGPT 3.5. And I actually bumped up to 4 this week, but it seems like it's kind of, it delays the text, like it's less chat. And I think it's more for just producing these, if you want medical images or those types of things, and that's a whole different field as well there. Okay, so this one in specific, so this is called text to image, and this specific model is done by a company called Runway. It's technically Runway AI. Stable diffusion is another type here too. So it seems like it uses some aspects of particles, and I believe that it's different than GPT-2 or GPT-based or BERT-based as well. And here's what I typed in: is a doctor helping a patient, and you can see here that they're photorealistic, they're clear. I think that's a huge hurdle. I've tried to do text to video, and it comes back as some Shutterstock. It's not generated, but I believe that these are, and it's trained on massive data sets, that Stable Diffusion first was trained on. And just by and large, so going from left to right on the top, it's usually in the hands, like the hands are just like, there could be 6 fingers, or there could be some kind of blending, that type of stuff. And again, I think that it's reached to the point where you could just keep clicking it 10 times and you'll see people post stuff all the time, no matter what, and say like, I generated this image. But doing it by code, I have access to the type of GPU. These only took 2 seconds to generate on an A100, like a 40 gig. And most of the time is actually in loading. So once the model is loaded, then you could just go one after another and generate images. So here's another one in the middle, doctor helping patient. And there's some things, some things with faces that, will likely get better, but it's usually those two. And then, so at first glance, you just say, oh, this is just, people in the hospital, that type of stuff. Okay, and then, so this next one is basically, so if you hear all of this buzz about explainability, and it seems to fight against, but it's not quite as strong, as the push for higher performance in models. Now, the push for higher performance in models typically means more neural networks or fully connected layers, self-attention mechanisms, those types of things if we're talking about transformers. So this is demo that I've been working on. I prove controllability basically. So if I increase the number of tensor networks, that it does, it's more controllable than the fully connected layers. So if I change the parameters, hyperparameters, or inputs. So this specific test is more about saliency. So saliency is a specific thing for explainable AI. And it's basically saying you can understand about or what the software is doing more based on these saliency values that are created. And what we see here is basically, so there's a chunk of code, and it's all up on the GitHub there too under explainability. And if there's a single tensor network, there's this much saliency. Now, when it's normalized to 1, ideally you only want one, your top saliency to be 1, and then you want all the other ones closest to 0, because it means that the computer is trying to focus in on that, or maybe select a few other ones that are like 0.6 or 0.4, whatever, 0 to 1, as opposed to trying to, because that's what it gives you insight into. And as far as interpreting what the computer does, if every value is just 1, right, that means that, it doesn't really understand the data set properly. And then, your next prediction isn't going to be much better, i.e. the loss of it all. Okay, so what was found here is basically increasing the number of tensor networks up to 4 decreases the total saliency. So I measured the median, the mean and standard deviation. So in all those cases, you want the smaller, the better. So it means that of all these, some value is going to be 1, and then the rest of the ones you want closest to 0, okay? So that, of course, would bring down the bars. Now, the other thing with the tensor network substitutions is that they're predictable. So you see this bar is this bar is greater than that bar, is greater than that bar is greater than that. So that's a really good sign. So, it means that as you're increasing the, the size of your neural network, or say potential, if this goes into transformers or an LLM, that this is looking, and people know that they're more explainable because this is linear. So the bottom one is nonlinear. And this data set, I put in about 480 points, which was about 6 times as much as the original model. And I also went further with some other data sets as well. But basically, if you look at the fully connected layers, you see how the bar lengths just vary drastically. And it's just known. Like, the inherent benefit with fully connected layers is these long range interactions. But the downside is the explainability. So you can see here the pattern goes away as far as, like, if you increase the numbers, it seems like it goes down, then it goes back up, and then it goes back down. And you can see the distribution of the bars are also, they more vary between blue, red, and yellow. So that's what was done on this one. And again, another way of doing this with saliency values. So these, again, I print out a table, I download it, it's like a CSV, and I can get my normal saliency values, which can range over 1. And then I can also normalize them to 0 to 1. Now, based on those same normalized saliency values from before, in this specific case, it's looking at the skewness of the distribution of these 0 to 1s, these, explainable parameters for machine learning. And basically, a high skewness means that the right tail is bigger, and then more of the data lies to the left, okay, meaning it's more 0, which is more preferred. And then for kurtosis, it's the peak. So a higher kurtosis is more of a peak, as opposed to a smaller kurtosis, which is more flat. Okay, so those two values, again, seeing a similar type of train, and this is supervised learning. This isn't yet into unsupervised with generative AI yet, but it's the architecture. So a lot of this should kind of carry forth. So you see that increasing the number of tensor networks is a similar trend as before, where lower was better. Now bigger is better. So for the lowest one, which is 4TN, 4 tensor networks, this is the biggest value in this predictable sequence. And you can see the same thing with fully connected layers from the previous slide, that they're just all over the place. And this is all, pretty much well known. As far as when you have more linear systems, and now the challenge is basically to get up to scale with tensor networks. And there's just a number of good papers this year that are basically putting tensor networks into the prime time. Now, this one should be the easiest to explain of the 3. Now, you've probably all seen model sizes, runtimes, and we know these trend together. Now, what's amazing about this is that for all 4 of the tensor networks is that their size is between 0.04 and 0.09 megabytes. So you can't actually see it on the first one, but if you look close enough, you could see on the 2TN, 3TN, 4TN. And then you can see, so red is the runtime and blue is the model size. So the fully connected layers are significantly larger. So we're talking like 300 times larger. And it's a small model, so this probably won't carry over as far as the 300x or the 350 times, smaller or less parameters. But just the, having that as a direct substitute, for other attention mechanisms, which has already been done in literature. And I'll show you 2 papers that they're already using it in transformers for LLMs. Okay, so you see this huge, gain in model size. And I think the best that I got on runtimes was my best tensor network versus this, say, 6 fully connected, was about 4 times faster. So the runtime isn't quite as pronounced as the model size or number of parameters. You're talking about, and this is all in charts and tables on GitHub too. You're talking about kilobytes of size, or 20,000 parameters for tensor networks, but you're talking about a megabyte or 2 on file size, and like 1 million parameters, right? So, this is very encouraging. So I'll keep going back to this, but basically, people want to take these LLMs or models that contain similar types of layers, like, fully connected layers, and then move them into cell phones as well. And this could have huge implications for medical. So this is one of the papers that I've been talking about here. They call it TQ compressed. So in this specific one, they took ChatGPT to small, and I believe this is up on Hugging Face. And then it's just substituting tensor networks, in this case, Kronecker decompositions in the embedding, multi-head attentions, and then the feed-forward network. And they got, pretty good results. And this one kind of stood out the most, this multiverse. So this is inside a llama. So this is another type of model. I think it's by Meta or Facebook. And basically, every time there was the transformer with the self-attention or multi-layer perceptron, substitute a MPO, which is what I was using a couple slides before, which is a matrix product operator tensor network. Now, in summary, where all this is going is, obviously, this has been a huge, long battle to get to this point. Chemical Q device, chemical equals biochemical generative AI drug discovery. Q equals tensor network, quantum-inspired technologies. Device is a anti-cancer medicine software development. So this is part of the cancer disease, medication strategy to do as much as this computationally. Are there any questions? And there was one in the chat here. Yeah, so Nupin said, I believe drug discovery deals with both the protein and the drug altogether. For example, docking a drug. Okay, where does the generative AI come into this specific thing with docking? It's something I've studied. I've taken biochemistry and medicinal chemistry and these types of things. I haven't come across it yet as far as how exactly. So what he's talking about is basically you have a protein and it's known to, dock in this specific site if it's a specific type of binding in that area. And to give you kind of a frame of reference, a lot of the code that I was getting was 2023 as far as generating a new protein sequence or doing a mask. Here it's called fill mask or mask fill. And so basically that is not too terribly old either. So I think a lot of these things, what you find up on Hugging Face is the general models. And there's probably people already working on that. If that, that's my guess, is to use Gen AI with docking as well.
Like my question was from a very simple picture that, when I imagine, my background is not in biochemistry. I'm a chemistry person, a computational chemistry background. So I deal with small molecules and chemical reactions and all that. But when we look at, drug discovery, we just assume that there is a drug and it's going somewhere and it's binding. And then I've seen some libraries that perform the, different combinations to see what fits best. So when we hear these words that, hey, this is AI for drug discovery, I just do not know, like, what's going on with the AI in the actual drug discovery problem.
Yeah, so the biggest thing is the Gen AI stuff, so more so the GPT models, as far as that's going to be things that generate, and it can't do every task that, say, like BERT can. So I would start off with those two. And then on top of that, I mean, if this is where you're heading to, is it's all based on the data that was trained on. So these, I think both of these are basically, it's a combination of, their amino acid residues and peptides or proteins, and then whatever the, GPT or ProtBert, was trained on, that's what's available to use for you. And I don't think that, say, for instance, that specific data set, and there's multiple data sets out there too, that I think it's more like this primary structure, where it's just like the code, that type of thing. So that's what you want to keep your eyes out for as well, because if you have a bunch of, if you have data sets that are based on molecular, binding and docking and these types of things, that's what it would need to be trained off of in order for you to now give it your data and now to get a high accuracy result.
Thank you.
Yeah, any other questions?
Yeah, Kevin, will we have access to the recording?
Yeah. I usually get it up pretty soon afterwards. And so it depends on where you're coming from. If you want more of the kind of drug discovery, if you want more architecture that's been covered in most recent lectures, like tensor networks. And there's also mixed in with like just the developments in, GPTs and that type of stuff.
Yeah, I'm more interested in drug discovery.
Yeah.
The use of AI in drug discovery. So do you have a YouTube channel, or where do you post? Oh, okay.
Yeah.
You put a link to that in the chat, please.
Well, yeah, it's just Chemical Q Device on YouTube.
Okay, great.
And then so you're going to see a lot more of this. Right now, obviously, because this just covered amino acids, proteins, but there's also DNA and nucleic acids, those types of things. But yeah, that's going to be the hard push with it all is drug discovery, and obviously the better technologies using tensor networks.
Okay, thank you very much.
Yeah, you're welcome. Anybody else that we haven't heard from? And then any other questions regarding the slides?
Hello. So I'm Abhinav.
Yeah, go ahead.
So, yeah, I think one of the biggest things that we will see in Gen AI when it comes to drug discovery is kind of this dynamic modeling of molecules. So just going back on what Anup was saying about you look at the interaction between the chemical moiety and the protein. One of the things that will actually eventually I feel will become important, which is still, even though we're at this Gen AI phase, I still feel like a far away to understand because we still don't have a full structured database of chemicals or of structures, things like that, especially when it comes to the 3D modeling. That's like one of the most challenging things. We will have the probably like the 2D structure or say like the smiles kind of like structure with just like the C's and the thing, but we don't actually know how a lot of the structures are. We don't have a database of like which angle are the bonds, what are the different rotamers that these amino acids can twist and turn, and how can the best affinity be kind of directed by these molecules towards the protein. So I feel like that's where Gen AI eventually is going to come in. What are your thoughts on that?
Yeah, I totally agree. So a lot of these things that I'm obviously secondary, tertiary, quaternary structure, those types of things as far as like what is the arrangement. So secondaries like alpha helix, beta folds, those types of things. If I were to hazard a guess, some of that might already been worked on today. Like I said, this is by the time they put all these up, it's all based on the papers really, because one of those slides that I showed before, and I said like the red dots are represent this as far as like that specific one is modifiable. It was either that one, they're those two papers, and basically they touched on it as far as like, it's based on the data set it was trained on. So whatever, it was like one was called a 50 and one was 100, and those are getting better as well. And yeah, I mean, these types of simulations, and even back in the day when I just did basic like chemical structures, so that are moieties of amino acids and like type of phenyl rings and that type of stuff, you could do that type of stuff in a very crude ChemDraw kind of way, or there was BioChemDraw and that type of stuff. But I think the answer is with the better data sets, and a lot of times you're going to have just a ton of data, requires more compute. So figure other leaders like NVIDIA, Google, these types of things are going probably way beyond the primary structure.
Yeah, no, that's a good point. But yeah, definitely I feel like because of the need of the data itself in some of these cases when it comes to the structures, I feel putting together the data first will be the big hurdle before we kind of are able to really move into using Gen AI to do drug discovery and like in our peak.
Yeah. Yeah, in my mind, I think it's a little bit ahead of what just an average person would think, because I have a couple degrees in chemistry. That was 2010, 2012. And I knew that these things, like, as far as like, one of them was using a specific data set or a specific modeling technique from back then. So, part of me thinks that if it was around back then it should be a lot better today as far as getting a lot of this, more of the information from it. Any other questions? And, you're going to see probably more breakthroughs from both, BERT-based and GPT. But from the sounds of it, and like this, definitely the headlines is it's primarily if they go past just saying we're just doing AI for medicine, they'll say like, oh, Stanford Medicine's using generative approach for drug discovery for this specific type of bacteria, that type of stuff. But I think that's where you're going to hear about it most, is the generative power. And again, if you're not coming from a strong machine learning background, it's just very simple. It doesn't require typically, to train on labeled data set like supervised learning. So this is all done unsupervised. And then, so that's synonymous with generative AI and that type of stuff. So we can't yet, there's not an FDA approved device where we can use unsupervised learning to take in a bunch of information and then generate an answer, because it's not that accurate. But we could use it for stuff like drug discovery, where we can, sift through just a ton of computational data to provide, good candidates, and then you can test further in the lab and then on your way to clinical trials and stuff like that. Any other questions, Anoop or anybody else? Deb?
Yeah, so I mean, my point is actually related to what I was saying earlier, and Abhinav was also saying that the generating all the molecules, all being like small molecules. I've seen a couple of papers that have like generative AI to just design, smiles formatted, strings, and they have like, pretty huge, like terabytes and terabytes of molecules. And then they can say that, okay, if you want to search anything, just use these, and do everything with these when it comes to drug discovery. But the problem is, because I'm from chemistry background and I do a lot of things within small molecules. The things we do even with ML are not that difficult nowadays. Now the graph neural network based potentials and the things that we do with them are actually getting very good, even for the unknown cases. So if we just know like a molecule is formed from carbon, nitrogen, hydrogen, and oxygen, any molecule that you form with those, we can give a reasonably good accurate answer with like a reasonable certainty. But when it comes to the drug discovery, the other part, the part where the protein is coming from, that is the challenge, because we cannot take the whole thing and do the accurate calculation. Now we need to either do active site approximation, just take the active site and then do it, or do something what you are saying today, that you generate the the protein itself. But I do not know how, which direction this thing is actually going, because we generally say that they have X-ray structures for the proteins that they are dealing with in drug discovery. So what is there to generate about it? So I'm a bit uncertain about that, but obviously it's a nice talk. I appreciate it. Thank you.
Yeah, I mean, if you're kind of talking high dimensions, that's kind of like the thing that tensor networks excels in. And so say, for instance, just traditional machine learning, if you do like principal component analysis or least, partial least squares, that type of stuff, it addresses it as well. But the thing you get with tensor networks is structure, like a specific, it's a 1D, 2D, 3D, whatever. And there's groups like iTensor out there that are, disproving, other leading types of hardware using this. And that's my initial thought is it's called high dimensional low rank approximation. So singular value decomposition, these types of things where it's a controlled approximation. So you're only keeping the data that is the most relevant. So if in that specific array or matrix, if that's the first, second, and third column, then instead of having all these other ones that are very low variance or significance, is that those get truncated. And I think that's kind of like the traditional approach of where these things are headed. If that's what you were talking about, is basically on the computational, we can only look at a specific drug, say, instead of saying, we want to see all these interactions going on, to a certain extent that we could with small molecules, but we can't with big because of the just computational processing.
Yeah. I mean, the thing you're saying about tensor networks, that can certainly help up to a certain extent. I am familiar with matrix product states or other type of tensor networks, and I do have actually a paper on it, how to do the tensor network based approximations based on graph theoretic decomposition of a biological enzyme. So, but what I'm saying is actually a little bit more complicated, because if you have, say, a few amino acids, then you can do these kind of things. But if you have the whole protein, then you don't know really how to deal with that other than molecular mechanics or some lower level of theory.
Yeah, I mean, the biggest thing that comes to mind is this iTensor group. They're based off Simon's Foundation, which is Berkeley, and then they're actually part of Flatiron. And in specific, they work on solvers. So instead of just doing everything by hand for they already have a solver for 1D matrix systems like MPS, but then they want to scale it up, and they say it's actually in development to now do like PEPs or tree tensor networks or Mera, things like that could more closely look like the structure of a big protein. So that's where a lot of the innovation seems to be. I could still do a lot of this, TensorFlow, matrix product operator, which is basically adding a leg to each node compared to MPS. And then in PyTorch or in Torch, you can create your own class, so like an MPS. But I think as far as like the more kind of complex systems that are taking like a lot of the to solve the bigger problems, I think groups like that's what they're working on. Because and even you can't talk about this without mentioning NVIDIA, because even on the quantum inspired side, is they've already simulated they said tens of thousands of quantum bits using tensor networks in specific. So for those of you that are less aware of tensor networks, basically the goal is to get whatever your system using the SVD that I mentioned down to like a matrix product state in 1D, which a lot of times that's really challenging to still keep the accuracy and do that. But you'll hear that a lot, like matrix product state, tree tensor network, Mera, those types of things. And it's simply a network of tensors. So if you know what a tensor is, like a multi-dimensional matrix, and then now you have a network, and that network, if it's in a specific configuration, is called MPS. And if it's in a different one, it's called a tree tensor network. Or if it's in a different one, it's called an MPO. And then each of those have their own defined sets of algorithms. So it's say, for instance, if you can get into MPS, that's the the fastest, most efficient sets of algorithms now, but you're in a 1D system now that was converted from a bigger model. So I think a lot of these things are definitely within the realm of getting it down into smaller systems. And what it sounds like is to have software groups like iTensor, where they work on these big solvers to basically now you can work on it without it getting like so complex, and you have to do everything kind of by hand here and there. So I think that's kind of where things are headed now, like with tensor networks.
Thank you.
Yeah, any other questions from anybody else? So, there's a good, so a couple of these codes, they actually came from the model cards on Hugging Face. So if you want to do it, actually, some of these things that were mentioned, like create a smiles from a small organic compound, well, you could do that like a primary sequence for amino acids into proteins, using the codes that I use and kind of develop further on. So that's available, but you have to start somewhere, right? So if you want to use Gen AI, or if you want to use GPT based or BERT, they kind of have their strengths going either way. But definitely it sounds like a lot of it's going into the generative nature of, say, we were trained on all this unlabeled data, and then now you give it a task, like create 10 new sequences that start with the code tryptophan. I think it's W, that one letter code, and then it'll develop whatever length of whatever, and it just gets all more precise and specific from there, like you need peptide size length or protein size length, these types of things. So, and then for everybody else, it's basically, I'd say that this is a really good area. I put up a poll as far as what everybody thinks is the best Gen AI, or what will be the biggest application in Gen AI for medical that will come out the door today. And I think it actually, there was drug discovery was over diagnostics, and there was clinical decision support, and then one other one. But so anyways, one of those other ones was the biggest one, but people come from all over. And I think it part depends on your background to, utilize what you're strong in too. And then anybody else that we haven't heard from? And again, I said, we got a link in the chat here too, another paper. And yeah, I cannot highlight enough as far as like when I talk about the databases that it was trained off of for like ProtGPT-2 or ProtBert. They're typically in papers, and then they'll compare it to the previous generation of databases or data sets. And then, your answers are like that 99% answer I got putting an X value in, when it should probably be 100% because it stands for any of them. That's coming from how well it was trained on that specific data set, right? So you could trace a lot of the accuracies and these types of things, back to the original where the big data set is, or where it came from. So yeah, there's a Science Direct paper in there too. So make sure to check that out as well. And any other questions, because what I found is you can't just say you're in medical, and I've tried other medical areas in years past, but unless you have the technical product, like especially with tensor networks, is I've gone like 10 weeks in a row on it, is that has to work in combination with a field like drug discovery, and then in combination with the main field, generative AI, right? These things have to work together, and meaning that each of these are kind of like the one to be in, and then you're like, oh, okay, this is here. But yeah, I would say it'll go heavily into biochem, if you get into it, and if that's what you want, then you should go for it, because that's where a lot of the research is pointing. And if any type of company, I would say you have to have something like a really strong assertion that this technical area that's not quite 100%, that's not quite incorporated into the mainstream. I'd say you have to have that too, right? So it's these things get built over time. And I'd also be interested, so I kind of got a sense of where kind of two people were coming from as far as kind of small molecule stuff and the challenges of going bigger, especially with the interactions. And, if you have any questions regarding any of the biochemical stuff, because what I showed you, that string, it's similar to like smiles, or actually smiles is another variation of one letter codes, is that's just primary structure. And then obviously the way it folds. So when you have a protein, it's never, it has these different, acid or base groups to them, amines and carboxylic acids on the side chains. So it's always going to be into some shape. Now, I think it was Anoop had mentioned this. X-ray crystallography is a key method that this is done in a lab, and it essentially freezes it, and you can see the structure of this amino acid, or and as far as I know, it's still not 100%, but I think for proteins, and you can correct me, I think there's MALDI is another one too, that's one of the leading ones is X-ray crystallography.
I mean, I personally am not a biochemist, but I think the accuracy of X-ray crystallography is pretty good, and we don't really doubt that side, because they have accuracy associated with the structure that is uploaded on the PDB, the protein data bank. Yeah, so that's, I guess, fine. Yeah.
Yeah.
The problem is like what to do with that now. Now we need to test out different things. That to me is probably the challenge. I am not actively working in drug discovery, but I'm trying to understand, because I do a lot of chemistry, like about 8 to 10 years of experience in computational chemistry. But when we look at these problems, this is a big problem now, no longer like small things.
Yeah. I mean, I guess my take with a lot of this is it was just all computationally impossible. Because like I said, if I took benzene and turned it into toluene, which is just a CH3 on a phenyl ring, then that's even more complexity on a laptop, So like I said, I would say a good starting point besides just literature, because that kind of gives you like data sets and the models available, is industry, is get up to date as far as like what specific tasks or hurdles were they able to accomplish. And of course, they probably won't share all the secrets, but that'll kind of give you an idea as far as like how much on software and hardware things have advanced, I guess, in more recent years.
Yeah, I will add that so cryo-EM is one of like the leading technologies these days in terms of kind of understanding protein structure next to X-ray crystallography. And so I feel like, right, if you think about anything, right, like even image analysis, for example, it depends on the resolution and quality of the image. The same way it depends on the resolution and quality of the structured data that we can get in order to train these models, so that we can get the best outcome when it comes to doing activities like this, like doing drug discovery with AI and other prospects like that.
Yeah, absolutely. Like I think the same thing too. I think even the like wet chemistry work that has to get better, and then capturing it into some image that's that always gets higher resolution, these types of things. And short of going down to the ocean floor, then finding out these molecules that are most resistant to obviously pressure or not having sunlight and these types of things as being good candidates. I think a lot of this, as far as like the X-ray crystallography going into PDB, that type of stuff, these exist in nature too. And, there's also like, so if you have a model that's like generative, so this GPT-2, which probably can be a lot more powerful than it is, it's just basically trained on what it previously was. So say if it even could do secondary or tertiary structures, your response will always be something that was based on all these other, databases or, experimental results, or just known things in physical chemistry or biochemistry, that type of stuff too. So I think there's multiple ways of looking at it. As far as like creating the next blockbuster drug doesn't, always have to come from, some other very specific, well, I could be wrong, I don't know, but I do think there's different ways of getting about using Gen AI, kind of making use of the tools that are available today. Anybody else that we haven't heard from? So again, this will all be up, and I guess, you know, for the startup or for the company, for me, it's going to be architectures. So it's like a GPT-2, I believe you could download that architecture and then, you know, modify it. And then, you know, in combination with, you know, just creating more and more useful software, you know, that gets into like secondary structure, that type of stuff. Because you hear about, you know, alpha helices and beta sheets a lot. Any other questions? And feel free, and if you could, I'm not going to pop that up because I got stuff on the screen. Abhinav, that specific paper, if you could kind of talk about it just in the chat, or you could come on as far as kind of like the nature of it. And yeah, so it's definitely a fun and emerging thing. And at the same time, you have to figure out what's currently being focused on as far as Gen AI with drug discovery there too.
Yeah, I think the main focus of this paper was looking at graph neural network
Oh, okay.
in order to kind of do drug design. This paper just came out this year, and so it was pretty exciting because it seems like they're paving one of the ways forward using GNNs in order to do drug discovery. And it's a little bit different from what you're discussing, but I feel that, you know, there are multiple different approaches that people can take with respect to these types of methodologies.
Yeah. And by and large, the two things that I hear that are resounding like on model cards on Hugging Face is just to whatever it is, if it's GPT-2, is then again to you know, fine tune it with your own data. So say like that was a good starting point of, you know, all these different sequences. And then on top of that, there's other, I don't know if you guys have heard of LoRa or DORA, which is like low rank approximations of the parameters. So you keep the parameters of this pre-trained model, and then you add like an approximated version of the parameters on top of it. And then so that's doesn't take as much as far as like compression. And, you know, it's supposed to help the original parameters as well. Because I looked up, you know, most of these, they kind of cap at 7 billion parameters. And that's like seems to be, you know, if you're getting 40 gigs out of an A100, something like that, with a data set that's like negotiable, as long as the data set's not too big. But you'll see some that are like 7 billion times 8. So that's 56 billion. And then you're not too far from like a Grok 1, which is like 300 billion. And then even farther than that, I think is like GPT 3.5 or 4. So anyways, on Google Colab, just by chance, and I don't think this will work for everything, but there's a 335 gigabyte TPU version 2 up there. And I would say the speed is probably about a low to mid-entry GPU, but the other ones cap out, like a V100 is like 51 gigs, and an A100, the one that I have, is 40 gigs. So anyways, I think that you're going to see a lot of these bigger devices pop up, especially on Colab, because, you know, even if, you know, everybody wants a bigger data set or trying to do more with it. And I don't think it's going to like cut it at 40 gigs. And they say an H200 is like 141. But these models, it's kind of leaving that area from like consumer, even though you're using industrial, like A100's industrial GPU, you're kind of leaving into, especially like with a Grok, like you have to have some more dedicated compute that you can get on Google Cloud. It's going to cost you, but you know, the TPU is like 10 cents an hour for this 335 gig TPU. And I've been using it on the models where the code is written for it. So yeah, so basically the huge driving force that I see is a lot with generative AI And again, BERT isn't typically associated with doing that. That's usually with GPTs. There's other types of GPTs out there besides OpenAI. There's Stable Diffusion as well, mixed up with that too. But that's where you're going to see, in my understanding, everything built around doing generative whatever in whatever field. So if this is medicine with drug discovery, that type of stuff. So, and the papers kind of support that. And definitely check out the industry news too, because NVIDIA has access to, just thousands of GPUs where we just have one. So it's just something to keep an eye on. Awesome. Any other last questions or comments? I appreciate everybody coming on. Go ahead, Noreen.
Yeah, I just wanted to say thank you, Kevin. This was extremely informative.
Yeah, you're
And
welcome.
I've subscribed to your YouTube channel and will watch some of those videos.
Yeah, you're welcome. Yeah, so everything kind of has to come together all at the same time. Any other comments or questions? Anybody? So again, this has been, let's see, Thursday, April 11th, 2024 for drug discovery with Gen AI, Tensor Networks, GPT, and BERT. Thanks everybody for coming on. Have a good rest of the night and have a productive rest of the week. Take care, everybody.
Thank you. Bye.
Yep. Bye.
Thank you.